Hello and welcome back to Biochemistry Pathway Series for Step 1, courtesy of 12DaysinMarch.com, where we'll be focusing on some of the biochemical pathways that you're most likely to see in your Step 1 boards exam. So for our featured pathway today, like all of our pathways, again our focus is not going to be on memorizing every single intermediate and every single enzyme. Rather, when approaching this pathway, we're going to be focusing on where our pathway starts, where it ends, what the goals or the purpose of the pathway is within the context of the cell, what are the key enzymes that are going to help us get from start to end, and what do they require to function, as well as key disorders that are related to each one of the pathways. And again, an overarching theme for our pathway series is integration of pathways, and how different pathways fit together to help us reason our way backwards and forwards through different problems. So for today's pathway, we'll be focusing on the process of nucleotide synthesis. This pathway starts with glutamine, aspartate, and CO2, and ends with purines and pyrimidines. As we mentioned earlier, the goal of our pathway is to synthesize purines and pyrimidines for the purpose of making DNA. Key enzymes in this pathway include UMP synthase and ribonucleotide reductase. Disorders associated with this pathway are hereditary orotic aciduria, but really when you think about dysfunction of this pathway, I want you to be thinking about chemotherapy agents or other drugs that are used to target this pathway. And as we'll see, this pathway fits nicely into pathways for folate and B12 metabolism, as well as the HMP shunt. So if we look at one of our integrated pathways, we can again see that nucleotide synthesis fits in nicely with the HMP shunt and the pathway for homocysteine elimination. You'll see the complete pathway for nucleotide synthesis to the left. For the purpose of today's lecture, we'll again be focusing on nucleotide synthesis, but I want you to note at the top and the right of the pathway that two of the key substrates for nucleotide synthesis come from the HMP shunt and the pathway for homocysteine elimination. So a major focus of looking at this pathway is going to be focusing on drugs that are specifically targeting to interrupt this pathway. And so the question becomes, why target this pathway with drugs in the first place? So if you'll remember, the fundamental point of this pathway is to make DNA for cells that are replicating. So if you target this pathway, you'll be specifically preventing cells that are replicating the most from doing so. So why do we want to stop cells from replicating? Well, in the first place, we want to target cancer cells. So tumor cells have rapid turnover and they're replicating out of control. So if we can prevent those cells from making new DNA to replicate, then we can theoretically target tumors. In autoimmune conditions, we have white blood cells that are replicating rapidly for the purpose of attacking whatever they're being sent to. So if we can stop white blood cells from replicating, we can theoretically dampen the immune response. A third target of drugs that interfere with this pathway are bacteria. So bacteria in the case of infection are replicating rapidly, and so if we can use antibiotics that target the nucleotide synthesis pathway, we can theoretically stop them from replicating as well. So before we delve in on this pathway, I want you to mention another pattern that shows up specifically in red blood cells. So again, you need nucleotides for the process of DNA synthesis. You have no nucleotides, you have no DNA. The nucleus needs DNA in order to mature relative to the cytoplasm. So what does this all mean? So regardless of how you go about interrupting this pathway, whether it be an enzyme deficiency, a vitamin deficiency, or with some of the drugs that we talked about earlier, if you can't make nucleotides properly, cells with high turnover will not mature properly. And for red blood cells, this means large cells, which is going to be described as having an increased mean corpuscular volume. So to break this down, if you interrupt any part of this pathway, you'll have increased MCV. So if you see this in a peripheral blood smear or on a set of labs that you get back, you need to be thinking about this pathway in particular. So this pathway is actually fairly cumbersome. So for the purpose of this lecture, we're actually going to break it into three separate parts. The first one is going to get us to UMP from amino acids. The next is going to make purines. And the last is going to use the UMP that we synthesized earlier to make pyrimidines. So each one of these aspects of the pathway is associated with different disorders and is targeted by different drugs. So that's what you want to focus on again when you're thinking about this pathway for boards. So let's look at part 1, which focuses on making UMP. As you'll see at the top of this section, we start with glutamine and carbon dioxide, which are two of our key substrates in the overall pathway. The first step of this pathway involves taking glutamine and carbon dioxide and converting it to carbamyl phosphate, using the enzyme carbamyl phosphate synthase 2. Now if you look to the side, you'll see that this process is actually ATP dependent and will yield glutamate at the end of this step. The next step of the pathway involves converting carbamyl phosphate to orotic acid, using the enzyme dihydroorotate dehydrogenase. You'll also see at this point aspartate enters the pathway, which is another one of our key substrates. As we'll see, the enzyme dihydroorotate dehydrogenase is a target of a drug called luflanomide. The final step of this pathway involves conversion of orotic acid to UMP by the enzyme UMP synthase. As we'll also see, this enzyme is implicated in a disorder called hereditary orotic aciduria. So I've just included this slide as a reference for luflanomide. As again I mentioned, I'll be talking about a few of the drugs that target this pathway, but I don't want to get into too much detail about any one in particular. But here you'll see I've listed some of the indications and adverse effects associated with the use of this drug. As I mentioned earlier, we can see that this drug is used for the purpose of immunosuppression, so the idea is to target rapidly replicating white blood cells for the purpose of suppressing its response in rheumatoid arthritis. So in terms of disorders that are targeting this aspect of the pathway, we mentioned hereditary orotic aciduria. So hereditary orotic aciduria is a rare autosomal recessive condition with a loss of function mutation in UMP synthase, which again is the last step in the process of UMP synthesis. So what does this cause? If you have this defect, children will typically develop mental retardation, developmental delay, increased MCV that we mentioned earlier, and excretion of high levels of orotic acid in the urine. So this can be treated by giving uridine, which essentially serves to bypass the missing enzyme and give you that UMP at the end of the pathway directly. So how is this going to present on boards? In boards, you'll typically have a child who presents with failure to thrive, developmental delay, and high levels of orotic acid in the urine. But again, if they do give you an H&H, I want you to look for the increased MCV that will trigger you to think of this pathway. In terms of question options, they'll usually ask you about the enzyme deficiency or how to go about treatment. So if we reference back to this part of the pathway, we can see that a deficiency in UMP synthase will lead to an accumulation of orotic acid, which will cause some of the symptoms that we're talking about in terms of mental and physical developmental delay. We can also see that giving UMP, or uridine, will bypass this enzyme, which is usually how we're going to end up treating this condition. So let's move on to part 2 of this pathway, which involves direct synthesis of purines AMP and GMP. Looking to the left of this side of the pathway, we can see part 1 that we've already discussed. So for the purpose of part 2, we're going to focus specifically on the right that begins with PRPP. Now an important thing to remember about PRPP is where it comes from. If you look at other lectures we have on the HMP shunt, you can see that PRPP is actually the terminal product of this pathway, which is how this process integrates with the process of AMP synthesis and GMP synthesis. Compared to part 1, this aspect of the pathway is fairly straightforward. First, you move from PRPP to IMP, which is then converted to either AMP or GMP, which are our end products. Now, each one of the steps in this pathway is actually listed as a single arrow, but actually involves a series of different enzymes. Now, there are a ton of different enzymes that you could try to memorize, but for the purpose of boards, it's not really important to remember them all. Instead, you can focus on the drugs that are targeting each aspect of this pathway. So for the conversion of PRPP to IMP, you target with a drug called 6-mercaptopurine. For conversion of IMP to AMP and GMP, you can actually target this step with ribavirin or mycophenolate. And again, I've just included a reference slide that talks about the indications and adverse effects of each one of these drugs, and you can see that they're either generally used for treatment of cancers or for the purpose of immunosuppression. So now we have our AMP and our GMP, and all we need to make are our pyrimidines. So for part 3, we'll actually be starting with UMP, which you can see that you get from the end products of part 1, and in one of the start products of part 2. So part 3 of the pathway is actually a bit more convoluted than part 1 or part 2, so we'll take a little bit more time to break it down. For the first step of the process, you can see that UMP is converted to UDP. From UDP, there's actually 2 ways we can go. The first way is to CTP directly. So from UDP, you can get CTP without having to use any other intermediates, and so you have one of our end products right there. The other direction UDP can go into is down to deoxy-UDP, using the enzyme ribonucleotide reductase. As we'll see later, this enzyme can be targeted by the drug hydroxyurea. Moving on, you can see that deoxy-UDP is then converted to deoxy-UMP. So I want to focus now on the last step of this process, which is conversion of deoxy-UMP to deoxy-TMP. There's actually a few things that we need for this process to work. First and foremost, you need the functional enzyme thymidylate synthase, which we'll see is targeted by a drug called 5-FU, or 5-fluorouracil. This drug works because it essentially mimics DUMP and binds irreversibly to the enzyme which inactivates it. So what does this enzyme actually do? So the role of thymidylate synthase is actually to reduce deoxy-UMP for the ultimate production of deoxy-TMP. But in order to reduce something, we need donators of reducing agents. In this case, our reducing donor is THF, or tetrahydrofolate. As you can see, tetrahydrofolate donates 2 of its hydrogens to DUMP to make DTMP, in the end producing dihydrofolate. Now the name tetrahydrofolate may sound familiar because you might just know it as vitamin B9. Rather than purely ingest more tetrahydrofolate for the purpose of making pyrimidines, your body actually uses a recycling process. So once you have dihydrofolate at the bottom, you can actually utilize the enzyme dihydrofolate reductase to directly generate more tetrahydrofolate for the purpose of working with thymidylate synthase. As you can see, this aspect of the pathway can either be broken down by having insufficient folate, or you can directly target the process of dihydrofolate reductase with drugs called trimethoprim and methotrexate. A final aspect to note about this part of the pathway is the role of vitamin B12. So when you actually consume vitamin B9, you consume it in the form methyl tetrahydrofolate, and as you can see over to the right, we've labeled tetrahydrofolate with a little methyl group attached. Unfortunately, you can't use methyl tetrahydrofolate for the purpose of TMP production. So you need a way of getting rid of this methyl group before it can actually be incorporated into the process. So essentially to create nucleotides, we need to get rid of this methyl group. And as we can see here, vitamin B12 is actually a key cofactor in the enzyme that serves to remove this methyl group, which will actually be used in the process of homocysteine metabolism, which is included in another lecture. But for the purpose of this lecture, I just want you to remember that B12 is needed to actually get activated folate. And here you can see I've just included another reference slide describing the drugs that are targeting part 3 of this pathway. So let's just take another quick look at all 3 of the pathways that we mentioned integrated together. As we can see at the top, the end of the HMP shun will ultimately help us get PRPP, which is a key early substrate for part 2 that we talked about. And looking at the bottom right, we can see that the removal of a methyl group from methyl tetrahydrofolate plays a key role both in the process of homocysteine elimination and in the process of nucleotide synthesis. So if when you watch these separate lectures you can reference back to this slide, it should help you reason your way through problems that are based on any one of these 3 pathways. Thank you again, and if you have any questions, please feel free to reach out to us at 12DaysinMarch.com.